Alterations in chromosome number occur frequently during gamete formation. About 5 to 10 embryos would have abnormal chromosome number, and indeed 50% of spontaneous abortion are due to such abnormalities. In some cases, abnormality in chromosome number produces offspring that can survive. In humans, you have trisomy 13, 18, and 21. Trisomy 13 causes Patau syndrome. The baby is born, but it has mental and physical deficiencies, wide variety of defects in organs, large triangular nose, and early death. Trisomy 18, you have Edwards syndrome, mental and physical deficiencies, facial abnormalities, extreme muscle tone, and early death. Trisomy 21, it is 1 out of 800, Down syndrome, mental deficiencies, abnormal pattern of palm creases, slanted eyes, flattened face, and short stature. For sex and chromosomal aneuploidy, XXY will form a Klinefelter person, sexual immaturity, no sperm, and breast swelling. XYY, you have 2 Y chromosomes, so you have Jacob's chromosome. You have Jacob's syndrome, so they are tall and thin. XXX, in females, they are called super female. They are tall and thin, and they have menstrual irregularity. They have XO, you have Turner syndrome. For Turner syndrome, you have short stature, webbed neck, and sexually underdeveloped. The autosomal aneuploidies compatible with survival are trisomies 13, 18, and 21. These chromosomes are relatively small. Aneuploidies involving sex chromosomes generally have less severe effects than those of autosomes. This is explained by X inactivation. Additional X chromosomes are converted into bar bodies. Some human aneuploidies are influenced by the age of the parents. Older parents are more likely to produce abnormal offspring. For example, Down syndrome, trisomy 21. The incidence of Down syndrome rises with the age of either parents, especially mothers. So, age of mother from 20, 25, 30, 35. At the age of 20 years old, the Down syndrome probability is 1 out of 1,925. 5 years later, it becomes 1 out of 1,205. And then at 30 years old, it becomes 1 out of 885. And at age 35, it is 1 out of 365. Okay, and then in ages 40 years old, it becomes a third compared to it, 1 out of 110. And 5 years later, it becomes even more, 1 out of 32. And at age 50, there's 1 over 12 chances to actually get a baby with Down syndrome. Down syndrome is caused by the failure of chromosome 21 to segregate properly. This non-disjunction is most commonly occurs during meiosis 1 in the oocytes. The correlation between maternal age and Down syndrome could be due to the age of oocytes. Human primary oocytes are produced in the ovary of female fetus prior to birth. They are, however, arrested in prophase 1 until the time of ovulation. As woman ages, her primary oocytes have been arrested in prophase 1 for a progressively longer period of time. This added length of time may contribute to an increased frequency of non-disjunction. Euploidy. Most species of animals are diploid. In many cases, changes in euploidy are not tolerated, and polyploidy in animals is generally a lethal condition. But some euploidy variations are naturally occurring, such as female bees. Female bees are diploid, male bees drones are monoploid, containing a single sex chromosome. Good examples of vertebrate polyploid animals have been discovered. In many animals, certain body tissues display normal variation in number set of chromosomes. Diploid animals sometimes produce tissues that are polyploid, so this phenomenon is termed endopolyploidy. Liver cells, for example, can be triploid, tetraploid, or even octaploid, 8N. Built-in chromosomes of insects provide an unusual example of natural variation in ploidy. In contrast to animals, plants commonly exhibit polyploidy. 30 to 35% of fern and flowering plants are polyploid. Many of the fruits and grains that we eat come from polyploid plants. In many instances, polyploid strains of plants display outstanding agricultural characteristics. They are often larger in size and more robust. Polyploids having an odd number of chromosome sets are usually sterile. These plants produce highly aneuploid gametes. Example, in triploid organisms, there is an unequal separation of homologous chromosomes, 3 each during anaphase 1. In this figure, each cell receives 1 copy of some chromosome but 2 copies of other chromosomes. Sterility is generally a detrimental trait. However, it can be agriculturally desirable because it may result in seedless fruits such as watermelon and bananas, especially triploid varieties. So, these seedless watermelon bananas makes it easy to eat, means that you don't have to worry about the seeds. But the thing is, without seeds, how can they reproduce? Because seeds are essentially eggs of organisms. So in this case, for humans essentially reproduce them by cuttings. So you just cut a banana tree, and then you plant it again, so it grows again. Second is seedless flowers. Seedless flowers such as marigold flowering plants, such as the taproot varieties. They develop by burpee, which is seed producers. Seed by burpee keep blooming since they do not form a desired. Seed developed by burpee keep blooming since they do not form the desired end product. And also, because there is no seeds, competitors cannot sell the seeds grown from their plants. Natural and experimental ways to produce variations in chromosome number. So there are three natural mechanisms by which chromosome number of species can vary. One is the meiotic non-disjunction, second is mitotic abnormalities, and 3 interspecies crosses. So for meiotic non-disjunction first is a failure of chromosome to separate properly during anaphase. It can produce haploid cells that has too many or too few chromosomes. If such gamete participates in fertilization, the resulting individual will have an abnormal chromosomal composition in all of its cells. So here, during fertilization, the gametes will produce an individual that is trisomic for the missing chromosome. The other one, these gametes will become monosomic for the missing chromosome. We have 50 normal gametes and 50 abnormal gametes. So figure 8.18 shows the type of zygotes that can result when the non-disjuncted gametes fuse with normal gametes. All of these offspring produced are chromosomally abnormal. So you have normal X, X, and Y. The non-disjunction one, XY, XX, YY, O, X, O. So as you can see, if a normal person has X, marries a person with Klinefelter, marries a person with a non-disjunction XY, you will have a Klinefelter person XXY. And then if he marries with somebody with non-disjunction of XX, you have triple X, super female. And if she marries a person with non-disjunction YY, then you will have XYY, Jacobs. If she marries somebody with a non-disjunction no sex chromosome, O, means that you have XO, Turner syndrome. Okay, so the other ones you can see that if the chromosome is from the man, X and Y. Okay, if the woman has the non-disjunction XX, you have XXX. If she has no chromosomes, you have XO. And if she has XX and that XX combined or fertilized with a man's Y chromosome, you have XXY. And then last one, if the man has Y but the woman doesn't have anything, O, then become YO. So YO actually results in death. In rare cases, all chromosomes can undergo non-disjunction and migrate to one of the cells. Instead of having two haploid cells, all of the chromosomes go to one cell and the other has nothing. Chromosome-less cell. It is a cell with no chromosomes at all. It is non-viable. So the red blood cell can participate in fertilization with a normal gamete. So this cell with all of the chromosomes can fertilize with a normal gamete to form a triploid individual. Mitotic abnormalities in chromosome can occur after fertilization. In this case, abnormality occurs in mitosis, not meiosis. You have mitotic dysfunction, chromatid separate improperly, which leads to trisomic and monosomic daughter cells. You have chromosome loss. Monocromatic chromatid does not migrate to pole during anaphase, so they just left behind. We have a normal and monosomic daughter cells. So in A, it is mitotic non-disjunction. They don't want to separate. So the sister chromatid actually brings the other sister into one pole. In B, chromosome loss. One chromosome does not attach to the spindle fiber, so during anaphase, they just left behind. So the genetic abnormalities that occur after fertilization lead to mosaicism. Part of the organism contains the cells which are correct and the other incorrect, which is genetically different from other parts. The size and location of the mosaic region depends on timing and location of the original abnormality, because you have some cells which are erroneous, some cells which are correct, meaning that the daughter cells of that incorrect cells will actually devolve or develop into organs and tissues and other cells. So you can see people who are mosaics. This is a fertilized Drosophila egg that is XX, but one of the X's are lost during the first mitotic division, meaning that during the original meiosis, normal, during fertilization, normal, but after it does the first mitotic division, one of the chromosomes lost. So you have a cell with XX, the other cell with XO. So this XX cell is normal. It will create a female fly. But the XO cell, it is abnormal and it developed into the male. So because of this, you can see that this fly would have both male and female bodies. It is a chimera. So this peculiar and rare individual is termed a bilateral gynandromorph. Bilateral means at two sides. Gene is female, andro is male, morph is shape. So it has half female and half male shape. So how does it happen? This is a picture of a lagging chromosome. So this chromosome, during metaphase, they are lined up. During anaphase, they are carried to each other's pole. But in telophase, one chromosome is lagging behind. They are too late. So at this point, the cell membrane, the nuclear membrane is being forming. And what happens when it is formed? When it is formed, the lagging chromosome can no longer enter. Okay, so due to that, only one chromosome enters the nucleus. So the nucleus will be made with one chromosome only. So the one that will be having one chromosome will become a male. The one that has full two chromosomes become female. This is what happens in humans. There are humans who are called hermaphrodites. They are an individual, not necessarily mosaic, with both male and female reproductive organs. Many sex chromosomal mosaics are known in humans, including the XXX, XYX, XX, XY, XXX/X. At least there is one case of a human XXXY chimera that results in fusion of two zygotes, one formed by a sperm fertilizing an ovum, and the other formed by a second sperm fertilizing the polar body of that ovum. So now you have somebody with both cells of a man and a woman. Aside from having an ambiguous looking external genitalia, that the person karyotype has both XX and XY chromosome pairs, and having both testicular and ovarian tissue. One possible explanation is a parthenogenic division of a haploovum into two haploova. So you have one ovum, the ovum suddenly divides into two. Okay? After fertilization of the two ova by two sperm cells, one carrying an X and the other carrying a Y, the two fertilized ova then fuse together, combine together, resulting in a person having dual genitalia, dual gonads, which is ovotestis, and genetic sex. There are fragile sites in human chromosomes. An intriguing discovery has been made in 1970 during observations of metaphase chromosomes prepared following human cell culture. In cells derived from certain individuals, specific area along one of the chromosomes failed to stain, giving appearance of a gap. In other individuals whose chromosomes displayed such morphology, the gaps appeared in other positions within the set of chromosomes. Such areas eventually become known as a fragile sites, since they appear to be susceptible to chromosome breakage and cultured in the absence of certain chemicals such as folic acid, which is normally present in the culture medium. Fragile chromosome made visible by atomic force microscopy, you can see there's a place where there is a small narrow path at the end of the chromosome. That is a fragile site. Clear association was established between several of these sites and the corresponding altered phenotype, including mental retardation and cancer. So again, Fragile X syndrome, or called the Martin-Weil syndrome. This disorder is named for an abnormal X chromosome in which a chip hangs on by a thin thread of DNA. This disorder affects one of every 1,500 males and one in every 2,500 females. Affected females usually carry only one fragile chromosome, which is dominant. Penetrance is not complete, and the trait is fully expressed only about 30% of fragile X-bearing females and 80% of fragile X-bearing males. In addition to mental retardation, affected males would have characteristic long, narrow faces with protruding chins, enlarged ears, and increased testicular size. The gene that spans the fragile side is now known as the one responsible for this syndrome. This gene, FMR1, is one of the growing number where the sequence of 3 nucleotides is repeated many times, expanding the size of the gene. The number of repeats varies within the human population. The high number correlates directly with the expression of the Fragile X syndrome. The normal individuals 6 to 54 repeats. Carriers, unaffected, 55 to 230 repeats. Fragile X syndrome, you have more than 230 repeats. The link between fragile sites and cancer. A link between an autosomal fragile site and lung cancer was reported in 1996 by Carlo K. Hubner and their colleagues. The F-HIT gene, standing for Fragile Histidine Triad, is located within the well-defined fragile site designated as FRA3b on the P arm of the chromosome 3. It is often altered or missing in cells taken from tumors of individuals with lung cancer. Extensive studies have now revealed that the normal protein product of this gene is absent in cells of many other cancers, including those of the esophagus, breast, cervix, liver, kidney, pancreas, colon, and stomach. Genes such as FHIT that are located within fragile regions undoubtedly have an increased susceptibility to mutations and deletions. Most recently, Muller et al. Have identified and studied another fragile site with most interesting results. One within the FRA16 site on the chromosome 16 is the WWOX gene. Like FHIT, it has been found to be either lost or genetically silenced in the large majority of lung tumors, as well as in cancer tissue of the breast, ovary, prostate, bladder, esophagus, and pancreas. When the gene is present but silent, its DNA is thought to be heavily methylated, rendering it inactive. Furthermore, the active gene is also thought to behave as a tumor suppressor, providing a surveillance function, recognizing cancer cells and inducing apoptosis, eliminating them before malignant tumors can be initiated. So, in a nutshell, F-HIT gene is the cancer killer. The WWOX gene needs to be turned on so that it can stop cancer cells from forming. If this mutation happens, WOX gene cannot be created, F-HIT gene is suppressed, then tumor can happen. Why? Because there is nothing killing the tumor by apoptosis. The end.